now we'll go from Australia to Germany. We welcome Bettina Lickiss. She is a very experienced scientist in cardiac field of iPSC-derived cardiomyocytes. She is scientific communications manager and business development manager at innovVitro. And now she will switch gears to a little bit different topic, and she will introduce us into the ways of assessing contractile force of human iPSC-derived cells. Bettina, welcome. You can start sharing your screen. Excellent.
Excellent. Well, already, hopefully already do.
Very good. Excellent.
Good. Going into presentation mode. Give me 1 second. Set up. So thank you very much, Elena, for the kind introduction and also the invitation to speak here today. I'm Bettina Lickiss, as you said. I'm business development manager of innovVitro. We're a partner company for nanotechnologies, and we provide FLEXcyte related services, especially for the cardiac safety field. So quite well fit here today. And I always start my presentations with a video where you can see the cardiomyocytes beating nicely because it's such a great eye catcher. But today I thought I'd make it a bit more interactive and actually make a little quiz out of it. So here you can see 4 of the wells of the FLEXcyte plate, and you, if you like to, can assign the compounds that you can see here on the left-hand and right-hand side to the respective wells. We also have the FLEXcyte signal underneath it to give you a little bit more of a hint. Put it in the chat box or tell me later. And of course, you will not get the results right now, because I'm going to show you a little bit more about the FLEXcyte applications that I want to introduce to you, the analysis of hiPSC-derived cell types, contractile properties, and after that, you will get the results. So stepping into the table of contents, because most of the projects I'm going to show you will be dealing with the technology, I'm going to show you a bit more about the FLEXcyte first, how it works and the parameters and etc. And then we dive directly into the robustness analysis of commercial human iPSC-derived cardiomyocytes, because we get that a lot from clients, that they're a little bit worried when they have to switch between lots. So they were not, is it right? Is it going to be changing the results, etc.? So we basically did an analysis of 10 different lots to see how robust they were. Then we're going to swap from this mixed culture cardiomyocytes to a different setup where we look at chamber-specific cardiomyocytes. So cardiomyocyte subtypes, atrial versus ventricular, do a contractile analysis of these cell types, compare them a little bit, also do a pre and post-compound analysis. And then we step away from cardiomyocytes for a moment and look into a smooth muscle cell maturation protocol that we established for, of course, also for using it for FLEXcyte analysis or contractile analysis. But because we use the primary cell, we have to work on the maturation first. So I'll tell you a little bit about that. And then last but not least, we were able to have the new Atlas device in our in-house lab, and we're a beta testing site and got some really good data that we would like to present to you. And after that, you will get the results of the quiz I just present to you. So starting with the technology, or basically why is it so important to have a physiological relationship for the cells? If you look at what we have normally in the usual lab, we have very, regarding mechanosensitivity, we have actually very extreme versions that we provide for the cells. On one hand, we have the regular cell culture plate, so very stiff material. The cells on top cannot really contract. They try, but they are basically too stiff to contract properly. So we have a maximum of stress here. On the other hand, we have usually a lot of cells in suspension culture, so the cells are floating and have nothing to hold on to. So we create a different kind of extreme for the cells, which they're usually not used to. And so they can contract easily, but they have problems relaxing anymore properly. So a different kind of stress, basically. So what we have tried with the FLEXcyte technology, with creating the membranes where the cells are on, basically from the video that you have seen on the first slide, is we create this physiological relationship that we need, that we also experience, that the cells would experience in the body, right? So with this physiological relationship, they have something to hold on to, but they also have the flexibility to contract properly. And this is something we have shown in presentations before already. This has a positive effect on the maturity level of especially human iPSC-derived cardiomyocytes. We get proper positive inotropic effects, etc., etc. Won't go too much into that, but very good for the cardiac safety community. So the technology here, this is one of my favorite videos because you can actually see the membranes very well. The membranes are of ultra-thin silicon substrate, basically, and they reflect the physiological conditions, as I mentioned. You can also see the difference here to the electrodes. The electrodes are actually not part of the plate, they're part of the device. And when the cells are on top of the membrane and they are contracting, you can see that they lift the membranes upwards and therefore create a distance change between the electrode and the membrane. And this is then measured via non-invasive capacitive distance sensing. So we have a label-free way to analyze contractility. And we also do it in a cell monitoring way. So we can do it acutely, but we also go up to chronic several days and measure it. And there's all the data we basically get is then analyzed quickly with a specialized software. Regarding the parameters, we of course start with one of the most important ones, the amplitude of contraction force. So we have a proper force measurement that we can perform here. We have also the possibility to go and measure area under curve, that we can dissect an upstroke and downstroke area under curve, because, you know, there are compounds that rather work on the contractile part or on the relaxation part, and therefore it's necessary to dissect these things. We can check the beat rate as well as the regularity or the irregularities, like the early after contractions that you can see here highlighted in red. So this is basically like a downstream physiological effect of an early after depolarization that will happen. And then we also look at the slopes, upstroke and downstroke wise, as well as the duration of the contraction, of course. And then here you can have any kind of peak width duration you want. So in total, we're able to really dissect the contraction into its bits and pieces with the amount of parameters we can measure. I always love looking at the raw data as well, because it just shows you the arrhythmia so nicely. So here we just have a simple control condition of one sweep. So basically a measurement of 30 seconds. And once you apply compounds that have an effect on the contractility, you see that we can see like beat rate variations, like highlighted in red here. We can see the earlier mentions early after contractions happening, highlighted here and with the red arrows, as well as proper arrhythmia events highlighted with the red arrows down here, when, for example, nilotinib is applied to the cells. Right, we got a lot knowledge about the technology now, so let's dive straight into the analysis of commercial human iPSC-derived cardiomyocytes. I already told you that the background of the experiments was basically to see how robust the different lots would react towards compounds, but also just generally speaking, pre-compound condition-wise. So what we did is we chose 10 cell lots that were produced in a time frame of 2 years. So not only like within a few weeks, but also like a quite long time span. And then we did a pre-compound analysis where we looked at the beat rate, and also did post-compound analysis where we checked nifedipine, so calcium antagonist, as well as sotalol, a hERG blocker. And starting here with the pre-compound beat rate, you can see very nicely that we have very stable conditions of the 10 different lots, with a beat rate ranging from 49 up to 53 roughly beats per minute. So we had a good start, and we then applied the compounds to the wells, starting with nifedipine. And you can see here that we have a concentration-dependent decrease of the amplitude upon nifedipine treatment, with high concentrations highlighted in pink and the medium concentration of 10 nanomolar highlighted in green. So the upper and lower lines here is actually the standard deviations that should be dashed lines, but the presentation mode kind of smudged it a little bit, sorry for that. But it actually shows that the standard deviation is below 10%, which is what we wanted to see, right? That there's a low variation, that this is a robust cell type that we can use for doing contractile property analysis properly. And the same was true for sotalol when we applied it to the cells. We have a very low standard deviation, again, below 10%, and we have basically no effect on the amplitude, but a concentration-dependent effect on the contraction prolongation, which is not really well visible on this slide, but then we try to depict it differently by plotting the different concentrations right next to each other also. And also this is just basically a downstroke duration shortcut. So here you can see that we have a concentration-dependent duration prolongation, which is basically representing the contractile response to the QT prolongation. And on the left-hand side, we have a similar effect of nifedipine, but the other way around, where we see the concentration-dependent shortening of the beat duration with a higher concentration shortening it. So in all, very nice data showing that there's actually no stress about different lots. You can use them all easily when you go for commercial cardiomyocyte. Next, as I mentioned, we looked at the chamber-specific cardiomyocytes. This is from a different commercial vendor. We treated them basically completely different, completely the same way. We added a 50K per well for both cell types. We had the same medium, did a pre-culture step of roughly 6 days, which is our very usual pre-culture step, and then did similar to the project before, like a pre and post compound analysis. In this case, we used carbachol and SBA. And here you basically have the pre-compound condition of the amplitude of the different cell types, and you can see already with your immediately the different shape reflecting to the different cell type, with a typical ventricular shape with the shoulder and the atrial shape here. Looking at the results of carbachol, which is an acetylcholine receptor agonist, so working predominantly atrial, so we were kind of expecting a strong effect on the atrial cells, which we also saw. We can see here that we have a duration prolongation, a strong one, a concentration dependent one as well, from 1 micromolar carbachol up to 100 micromolar carbachol. And for ventricular cells, we see a similar effect, but just much smaller in comparison to the atrial ones. Another compound was tested, was the SBA one on ventricular and atrial ones, where we had the control condition here. And what we can see here very significantly is just 1 minute after applying the compound, which is an L-type calcium channel agonist. So we have a strong increase in the duration for both cell types. And what then is very interesting to see is that after 10 minutes, we have a very physiological effect of SBA on ventricular cells, where we see an increase in beat rate. So all in all as well, this was a nice project to show the differences of the different subtypes of the ventricular and atrial cardiomyocytes using contractile property analysis. Okay, leaving cardiomyocytes aside for a moment, we go to smooth muscle cells on the FLEXcyte 96. And the next cell type we basically want to evaluate, or are evaluating right now. This is like the first few projects that we did for it. And because we don't want to go straight with commercial cells, we were using a primary human aortic cell line. The advantage is obviously that we can just grow it in-house and have up to 15 population doublings. The disadvantage is that because they're proliferative, they're also kind of non-contractile, so we had to work on the maturation protocol first. And that's what we did. We actually fiddled about with quite a few different setups, and was always like kind of having them in culture for 7 days before we then plated them onto the FLEXcyte plate, and had half of the plate with maintenance medium and the other half of the plate in like changing maturation style medium, looking for which one would be the best one for doing a functional, for the readout as having a functional analysis with the FLEXcyte, as well as the phenotypical analysis where we did some immunostainings. And the ones that worked best actually for us was where we had basically a reduction of everything in the medium, where we would go from 5% FCS down to 0.5% FCS, and also basically got rid of the growth factors completely from EGF and FGF. And that seemed to work out very well for the maturation of the smooth muscle cells, as you can see here with the phenotypical analysis. So on the right-hand side, we have the maturation medium, and the phalloidine staining, basically a staining for actin filaments, shows actually very nicely that we have an organized cytoskeleton in comparison to the maintenance medium here on the left-hand side, where everything seems to be still a little bit disorganized. So that was a good step forwards. And then we also performed some functional analysis where we used phenylephrine, so an adrenoreceptor agonist, for checking basically the contraction behavior. And you can see here very nicely that we basically, with the blue bars, we have no response in the maintenance medium of the smooth muscle cells that were in the maintenance medium, but the ones that were in the maturation medium did actually react to phenylephrine quite nicely, with a almost 30% increase in force of contraction after the maturation process. So here I think we found a good way of getting our protocol into place for doing follow-up studies, taking more compounds in and checking the smooth muscle cells more regarding their contractile properties. Right, our last project actually is the new product of nanotechnologies. So that's what Sonia introduced in the morning. We were happy to be beta test site for the Atlas, and were able to have to create some data very quickly thanks to the amounts of wells that you can basically analyze at the same time. Because as Sonia mentioned already, we have the possibility to actually test 96 well plates at the same time. And this has nothing to do with contractility, by the way. This is an impedance-based system, and the frequency spectrum here is actually quite broad. There's a lot of things you can analyze between less than 1 kilohertz and up to more than 40 kilohertz. So just to pick a few, there's the teal mode, for example, interesting for hepatocyte analysis, and cell monitoring mode. That's what we did for our cardiotox study, and cytolysis mode, for example. So the study design was to test chronically the cardiotoxicity for 3 days using the cell monitoring mode. And because there's so many wells and there would actually burn quite a lot of commercial cells, we decided to go for cell line again and use the H9C2 cell line, so red heart myoblast cell line. That has like acts similarly to the smooth muscle cell line that we use. So they're proliferative, which is great because we can increase them quickly, but also they're rather immature. So we had to use serum salvation again to induce the myotubes. But we were successful with doing that, so that was actually no problem. Here's the myotube, for example, and this cutout here shows that we even have multiple nuclei, and therefore, we were basically ready to go, plated them on 696 well plates, and basically performed this study within a week only. That was quite cool. And the compounds that we selected were actually a subset of the HESI cardiotox study that was just being performed. And that was basically chosen because we had so many different mechanisms of actions here that were basically working on the cells. When we were therefore testing a broad range of ways of cardiotoxicity very successfully, actually. So I won't go into detail with all the compounds. I'll just show you a few of them, starting with pentamidine, for example. So here, what you can see here is the cell signal rising is when the cells are getting confluent on the plate and basically impeding current. But if you would apply a compound that would have a structural cardiotoxic effect, then you will see a decrease here. In the case of pentamidine, you don't see that, which is actually a bit confusing because usually you would see that at least 24 hours after administration of the compound. But because the cell line has got no hERG, of course, you won't see any hERG trafficking block. And that's why it's actually a good control that pentamidine is not showing any results here. A proper control is allotinib that I would like to show you here, where we have strong ranging concentrations from 230 nanomolar up to strong 7.7 micromolar concentrations. And still you have no strong cardiotoxic, or actually no cardiotoxic effect, which you can also see here on the analyzed level. It's a nice cardio-safe compound showing no toxic effect on our cardiomyocytes. Now it's getting a bit more interesting. We have a cancer therapeutic agent here, vincristine, and we see concentration-dependent decrease once we added the compounds. So basically an increase in the toxicity that is also visible here when you look at the analyzed data. And that the higher the compound concentration is, the more toxic it gets for the cells. And the similar effect is visible very nicely on a concentration-dependent level for doxorubicin, of course, and anthracycline that is heavily cardiotoxic. And you can see here as well very nicely the concentration-dependent decrease in cell signal, basically meaning an increase in toxicity. And that was, to wrap it up, like I said, very nice to perform this study, especially because it was done in no time because of the throughput. And this, I would like to thank our partner Nanotechnologies just for great partnering in general, but also Fujifilm and Axel for awesome collaborations that we perform. And of course, last but not least, the FLEXcyte quiz. I hope you put some of your thoughts into the chat, because this is the result. We have isoprenaline on the left-hand side and the control condition coming after that, SBA on the right-hand side, and nifedipine on the far right. I'm sure most of you are right. Well done, you. And I'm open for questions.
Thank you very much, Bettina. Very nice talk. I was about to remind you, where are the quiz results? We already have some questions for you. So the first one came from Martin Trebert. Can you co-culture the smooth muscle cells with endothelial cells?
Well, this would definitely be something that would be a follow-up, because that was like an initial study. You know, we wanted to get everything in place, but yes, perfect question, and will be noted down, and we'll probably be able to present something like that in the next cardiac safety meeting.
I have one hand and one question written. Maybe I read first, and then Marcus, I let you sign in. So from Puya, I hope I pronounced that correctly. To evaluate the drug-induced changes in cardiomyocytes, the PASHCON technique can measure the cardiac action potential in all the individual ion channel currents. Then how would the measurement of cardiac contractility be important for preclinical drug toxicity screening?
I think it is always important, because what we do in preclinical drug development screening is we look at actually the three of them. We look at the electrophysiology, we look at the calcium transient, and we look at the contractility. And personally, I think the contractility gives you the best view, because the contractility is at the end of the whole contractions of this whole motion of cycles. So whatever you see in an electrophysiological level, you see as a contractile answer. What you see on calcium level, you see on the contractile answer. The other way around wouldn't work. If you have an effect on electrophysiology, but you, for example, would have that's fine. If you would have an effect on calcium, you wouldn't see that if you only do patch clamp. As of course you would see that too. But it's the best way basically to start it from the back, because then you can include all the others as well. So this gives me the that's why I think this is the most important way, to start basically from the last bit of the contraction cycle.
Or one does both.
Yeah,
that's also an option. Marcus, can you turn on your microphone and ask your question?
Hi, Birgit.
Bettina.
Hi, Bettina. Sorry, very nice talk. Sorry.
No
I have a question. Can you estimate if the layer of cells, is that really a monolayer around in the FLEXcyte, or would you think it's more like a three-dimensional shaped orientation of the cells?
It is more monolayer, because we always do an evaluation of the cell type, no matter which one it is, and then we choose basically the perfect conditions where we have the right amount of cells to get a confluent monolayer, but also not get too thick. And also just to basically save cells, because especially the commercial ones are, you know, not can be a bit expensive. So you kind of have to find the right way in the middle. So what we present is monolayer.
Okay, thanks.
Thank you.
Thank you very much, Marcus. So more questions, really nice. Takasuna-san is asking if you can co-culture smooth muscle with endothelial cells. We expect that you will evaluate the acetylcholine-induced EDRF effects.
Okay, well, that sounds interesting, but we haven't done that yet, so I can't answer that, but can't comment on that. But it's a good hint. Thank you very much.
And the last one from Adam Hill, nice drug data on FLEXcyte. Are you aware of any data showing differences in contractility as measured on FLEXcyte for disease-causing variants versus normal cardiomimes?
We do have projects going on for that, but at the moment I wouldn't share that right now, but maybe in the next, for the next safety meeting, I would. Thank you.
Thank you very much, Bettina. So we're perfectly on time.
Perfect.
And very, very nice talk. Thank you so much.
Thank you too.
We will switch now to